I just want to introduce the team tonight. So Dr. Gretchen Hofmann, I met a long time ago. She and I go way back, and she was up here visiting me at one point in the last year and came in and saw this incredible space and was like, wow, we should do something up here. And I was like, well, yeah, we should do something up here. And so she and I kind of schemed on it and came up with this sort of idea. And she's come all this way from Santa Barbara. She has brought her entire lab group, who are the people in the red shirts back here. You guys can raise your hands. These are all grad students in Gretchen Hofmann's lab. And they're all people that work on various projects, some in Antarctica, some in Santa Barbara, but they're the ones that are really the backbones behind all these great activities you got to do and trying on the gear and the postcards. And so if you haven't done a penguin postcard and you want to do one, catch one before you leave. And then I also want to introduce Lily Simonson, who is part of the show tonight. And she is, she's an artist, and you can see her beautiful painting up here. You'll see a lot more of her work in the presentation tonight. And so the theme tonight also is sort of like using art and science as a way to sort of bring more people into the ideas that Gretchen will be talking about, and Umi and Lily will be presenting on, is just how our environment is changing and the kinds of science that they do. A lot of people have a connection with art, and I think this has been a really successful collaboration with them. So I'd like to just read off a little bit of their bios before we begin. So Gretchen Hofmann is a professor of marine biology at UC Santa Barbara. She conducts research both in the Antarctic and the kelp forests of California. And Lily Simonson is an artist who embeds in research expeditions. Her works have been exhibited across the US and Europe and has been featured in outlets such as the Los Angeles Times, Smithsonian Magazine, MTV. She holds an MFA from UCLA and a BA from UC Berkeley. And Umi Hoshijima is a PhD candidate working with Professor Hofmann at UC Santa Barbara. He has conducted 3 seasons of Antarctic research and focuses on how organisms interact with local seawater pH. So without further ado, I'm going to hand it over to Gretchen.
Thank you, Carol. Thanks so much. It's a pleasure to be here. How's the sound back there? Is it too loud? Is it okay? Right on. I'm wearing my very first Madonna mic, so we don't know what's going to happen here. Feel a little insecure. Oh, I did that earlier. We rapped. We might rap when this is over. Umi's got some mad rhymes. He's like, no! Well, thank you for coming. It's an incredible pleasure to be here. We are all an incredible mixture of diversity and different kinds of talents. Some of us are molecular biologists, others are divers or ecologists, but we're all really committed to understanding how organisms will respond to the coming climate change that we expect on the world's, in the world's oceans. And tonight we wanted to feature some of the work that we've been doing in the Antarctic. And the Antarctic is one of the parts of the world's ocean, the Southern Ocean, where we think it might be the most vulnerable to a phenomena called ocean acidification. And Umi's going to illuminate that for me later. But what we're going to talk about tonight is a beautiful little harbinger of change. This is Limacina halicina antarctica. It's a teeny tiny pelagic snail. I don't know, a whopper is like the size of a lentil maybe, and little ones are the size of a pepper grain. And we know that they're found in all the oceans, the Antarctic and the Arctic, and they're incredibly important food in the food chain, especially for fish. So what I'm going to do is open with the rationale for why we study something this crazy. It's got this beautiful shell that we actually think is very vulnerable to ocean acidification, such that juvenile forms in the future might not actually be able to calcify, which is bad news. But the good news is recent results have told us that organisms are kind of tough, right? So they're plastic, and they have capacities to adapt to change. And one of the things that we study literally is, do organisms have the plasticity and the capacity to adjust to environmental change in time for us to get our act together and stop changing the pH of the ocean? So, thank you. No, it's really important. And the thing is, I think we can do it. I don't doubt it. We just have to focus. So what we're going to do is talk about this little guy. Umi's going to talk some about the science. I'll introduce it. And then Lily will close with some beautiful perspectives on how art informs and magnifies the science. So here's our pal, the sea butterfly. And it's really it's a snail. It's a marine snail. And instead of having a creepy crawly foot, evolution has modified the foot into these beautiful swimming paddles called parapodia. This is a juvenile where their wings look more like paddles, and as they mature, they start to look like Dumbo the flying elephant. The paddles get really big. And in the ocean, they're very patchy, and they occur in clouds, and they are part of a really important part of what we in the ocean call the marine trail mix. This is me. Because out in the ocean, there are tons of little crabs and crustaceans and copepods and all sorts of things that are part of the food chain. They might graze on plankton, and then they themselves are eaten. And they're incredibly important, and we don't always appreciate the role that these things play as a linchpin in marine ecosystems. So one of the things that we learned when we wanted to study these guys, because they're so vulnerable to ocean acidification from the shell, is that when we captured them, they're actually fairly athletic. We get them with plankton toes. Umi will show you, and they're tiny, but they're like, they swim up and then they go woo, and they come down, and they swim up and they go woo, and they come down. And this is what they do in nature. Some of them vertically migrate following the sun, but they spend a lot of energy moving around, and they appear like this in nature, and fish will come in and eat them. So one of the things I wanted to show you is some video shot by another artist, Henry Kaiser, who's an underwater videographer. Ums can turn up a little bit. And Henry's also a trance guitar player. He's quite famous, actually. So he laid down a little music over this video. And these fish are cryopelagic species. They live in direct association with ice that would freeze most everything else. But they have, oops, we lost it. They have antifreeze in their proteins, in their blood. Let's try it again. Let's play it again. So, and what these guys do is they eat the Limacina. And some of the research done by New Zealand scientists has shown that 80% of the gut content of these fishes are pteropods. So we've got the fish eating the teeny tiny pteropods, but to keep going, what eats the fish? And this is where the fun starts, because when we work in the field, this is us on a routine day. We're out at the ice edge, and these emperor penguins were out fishing, and you can see a bunch of them in the water diving, and they're eating fish diving under the sea ice. But because there are no predators on land, they just came up to check us out. And they're about this tall. And so I'm just standing there with my camera. And that's a really typical call. They're sort of checking you out and communicating with each other. And these are actually juveniles. This time of year, they should be breeding out at a rookery. So these are actually juvenile delinquent penguins running around together in little packs feeding. So they're teenagers, and they are really curious. We've had them come up and vibe our vehicles like, ugh, bah, me. So it's actually very funny. But they're very bold. They are not afraid of us at all. But we are extremely boring, and they've decided eventually that we're not worth hanging out with.
How tall are they?
Yeah. And there they go. And see them toboggan, and their feet are incredible claws, so they get traction on the ice. And boo! But you can see their brothers out there in the water, and they're swimming along, and this ice is probably like 6 to 10 feet thick, and they're diving under it to get fish. So the pteropods feed the fish feed these guys. So what we end up with is really a very short food chain, if you will. This kind of food chain is probably one of the shortest that we know of, and not only do the penguins eat the fish, but the seals do as well. So the zooplankton, the Limacina, is a really important part of what makes these Antarctic ecosystems tick. So our goal was to go down there, do a little science, collect these animals, and in many ways we wanted to study their shells, we wanted to study how they work, but we also want to know what will happen to them in the future. And in order to do this, we have lots of different moving parts: diving, field work, lab science. McMurdo Station, where we work, a US base, has beautiful lab facilities, so we're incredibly privileged to go there and work. So I'm going to hand it over to Umi now to give you some science.
Hi, can you all hear me? Great. So, I'm going to talk a little bit here about ocean acidification. So Gretchen mentioned this a bit just now, and this is something we're really concerned about in the near future. So, as you may know, we're emitting a lot of carbon dioxide into the atmosphere. This carbon dioxide gets dissolved into the ocean. So about 30% of the carbon dioxide we as humans have released into the atmospheres has actually been already taken up by the oceans. And that might seem like a great thing because, well, it's not in the air anymore. The oceans have sort of taken care of our problem a little. But it turns out that when carbon dioxide goes into water, it forms an acid called carbonic acid. And that's that in magenta right there in the middle. And of course, when you add an acid to water, it lowers the pH and makes it more acidic. One other consequence of carbonic acid is because of the way the chemistry balances out, it turns out it reduces the amount of carbonate, which is that green guy right there. And carbonate's really important for a lot of marine animals, because it turns out that a lot of the shells of marine animals are made out of calcium carbonate. So they really need a good amount of carbonate in the water in order to make their shells and to have them hang around. So what we're concerned about is as there's more carbon dioxide in the water can eventually become too acidic to the point where animals either find it hard to make their shells, or the water can actually start dissolving already existing shell, and the animal may have to fight in order to keep rebuilding it. So that was my chemistry slide. That's out of the way. And so I'm going to talk a little bit about our whole research project right now. And so we sort of have a few different parts of this. So we're really interested in pteropods as a sentinel species for ocean acidifications. We know they're globally distributed and they might be vulnerable to ocean acidification, and they're really critical in a lot of different marine communities around the world. So we know that shell dissolution, shell dissolving, is caused by low pH, and we can actually check that out by putting these animals in a very accurate microscope and a scanning electron microscope. Now, in order to sort of pair that with some laboratory experiments, we can make future oceans in the lab. So we can take carbon dioxide at different percentages and mix them with seawater and see how pteropods do in different future conditions. And in order to figure out what the future conditions are, of course, we need a really good grasp on what the current conditions are. So the third part of our project is to put pH sensors in the water to look at how the acidity of the water changes naturally year-round. And from taking all that information, we can hopefully get an idea of how the pteropod is doing now and how the pteropod will be doing in the near future. So I've had the privilege of going down to McMurdo Station 3 times. I just came back from my 3rd trip. Each time I go about 1 to 3 months. This is the station. It's 1,000 people maximum capacity. It's the biggest station in Antarctica. That blue big building is our galley, our dining hall. All of those at the bottom are dorms. There's another building in the middle right there. That's our laboratory facilities. And so the National Science Foundation has a lot of infrastructure in place to help us do our science. So it's an incredibly humbling place to do research. And it also gives, personally me, a lot of responsibility in making sure that I get my science done and with all this wonderful support around me. And what you can't see in this photo is right here on the bottom right. That's the coastline right there. So this is some premium coastal real estate right there. And we're right on the coastline. And when we go there, we want to go in about the spring to early summer, because in the winter it's too cold and too dark to work. But in the summer, it's too hot, and the whole ocean in front of us melts away. And we rely on the frozen ocean right outside of station so we can drive around and get to all the places we want to go. So we use these snow groomers to get around. They're sort of our big minivans that we can putt around in. You can speed around at 10 miles an hour and get wherever we need to go. So they're really great in that we can take, a good shelter with us when we go out to do our work. So as I said, this ocean is covered in frozen ice, so about 6 to 10 feet thick everywhere around us. So you got to get through that in order to get to your animals and to get to the seawater. So the way we do that is with a video. Specifically, a video about... So this is one of the big drills that they drag out for us. I said station has a lot of equipment and infrastructure for us. So they have these bulldozer drivers that will take these massive drills out and drive them out to our research sites. So that drill bit is 4 feet in diameter, and I believe that's a machine usually used for drilling big fence posts and telephone poles. But they've rigged it up so it can drill holes in this really hard Antarctic sea ice. So there it is, getting moved into position. And then it just starts grinding away at that and slowly chipping away and slushing that away. And then about 6 feet of grinding later, you get a perfect, nice cylinder all the way down. So it's really incredible that, this ice is so thick you can drive a bulldozer over it, and we can just get down to the ocean from wherever we go. And being in marine biology, you're so used to taking a boat somewhere you need to go. If you're diving, you might have to swim out from the shore. But just driving out in your minivan right up to the spot where you're going to hop in is such a crazy experience for me. And then what happens is that's a hut that we can drag over the hole. And what we do is we take this nice heated hut that we call a fish hut and drag it over that hole. And these fish huts are great because if you just left that hole there, it'd freeze over, as you guys would probably know, really quickly. And so if we wanted to go back there multiple times, we'd have to chip it out or drill it out every single day. By keeping this heated hut over the top, it keeps that hole open and also gives us a nice, sheltered, warm place to work out of. And this is one of the ways that we collect those pteropods. So we take this big net called a bongo net. And it's called a bongo net because it's kind of like 2 drums put together. It's a really goofy name. But what that thing does is we drop it down our hole and we leave it hanging there. And it sort of swivels with the currents like a sail, and it catches anything that swims by for about 24 hours. And then we pull it up, and we have ourselves a bunch of pteropods, hopefully. And it can be a little tricky getting it up the hole sometimes, so that that bongo net is just about the same width as the hole. And as we have the hole open longer and longer, it gets narrower and narrower as it sort of freezes back. And so it's sort of a little challenge sort of getting it back up, but it's always fun. And then what happens is anywhere where you have a good big hole that you keep open, a seal will come and breathe out of it, because these seals are living under the solid ice and they really need these nice places to breathe. So we get visited by these seals very regularly, and you learn to sit back and let the seal do its thing. And then when it goes diving, you go really quickly work. And then you see the seal come and you back off. So you sort of get in a rhythm with the seal, which is really fun. So once we got these guys in the lab, one of the things we were interested in is looking really closely at their shells. So you can see from the bottom that that's a millimeter. So that's about an average size shell we were getting. It's about maybe a little more than a millimeter. And that looks pretty good, but we can actually zoom in even further. So this is really, really small. And just looking at that very middle, this very center whorl of the pteropod shell. And you can see when you get this close that the surface is not just this perfect smooth area, that some parts will start to look a little rough, almost spongy. And that's actually the crystals of calcium carbonate underneath the top protective layer. So that top protective layer is gone in some places, and you're actually seeing like the framework underneath. And what we can do is, after some visualization, we can start to categorize the conditions of different parts of the shell. So one might be a perfectly good layer of the shell, and a 2 might be a little roughed up like that. And then a 3 might be like you see in this photo, where you start seeing more and more dissolution and really getting into the crystals in the shell. And we think that this sort of pattern of dissolution could start to become damaging to the shell, and the pteropod may have to potentially use more energy in order to constantly repair this really essential part of their body. So that's sort of a quick overview on this first bullet point of how we've been looking at these pteropod shells. And I mentioned that we sort of make future oceans in lab. So it's this kind of crazy system of Home Depot parts, which really is, a big part of what we end up doing. So it's these 5-gallon plastic buckets in the middle is where we mix different amounts of carbon dioxide into seawater. And then that gets pumped up into all those small tanks around the outside. And that's where we hold our pteropods. So we're able to make different treatments of carbon dioxide and put that into different tanks in order to hold pteropods at different pH, different acidity, to mimic different conditions in the current and near future. And just by looking at them under a regular microscope, not even a light scanning electron microscope, you can start to visually make out some cracks and pitting in the shells in high CO2, so low pH waters. So this is the kind of water we might expect to see at the worst time of season in about the year 2100. So in about 100 years, at the lowest pH of the year, we expect it to be about this bad. The top left is sort of the best part of the year right now, so the most favorable water for shells today. And that top right is sort of the worst, lowest pH that you might expect to see today. So you can see that as you go gradually into higher CO2, you start seeing more and more visible pitting of the shell and potentially even some cracking as well. So finally, one really exciting part of the research that I've been heavily involved in is deploying pH sensors. And after all this exciting biology, a pH sensor might sound a little boring. You've done that in chemistry class. You dunk it in a beaker and you measure the pH. What's the big deal, right? Well, the great thing about this is that it's been able to get me diving down there. And the diving in the Antarctic is possibly the most phenomenal diving on Earth, and which means that I've already peaked, and I don't really know what to do now. I should go dive the tropics, but the water visibility is beyond clear. You have the ceiling above you of frozen ice, right? And as the seasons progress, you'll get some algae on it, so you have this green ceiling above you, almost like a big stained glass ceiling above you. This is actually a contraption they put under the ice. So those are squares at the top are windows, so you can actually climb down a ladder and see what's under the ice without getting wet. So it's for people that aren't divers to be able to experience a part of what we were able to do. So of the 1,000 people on stations, I'm one of the lucky maybe 10 or 12 or so that actually get to go dive in the water. So it's an incredible privilege already to be down there as a researcher, but the ability to go dive down there and experience this is a whole nother, has been just completely phenomenal. So what I do is I do a lot of wrenching underwater and electrical taping. It turns out that it's not as glamorous as it sounds, but what we do is we swap out these pH sensors and try to keep a long record of what the pH does down there over the seasons. And what we found over a number of years is those blue dots are the average pH of every month. So when we go there in November, the pH is about a regular 8.0. That's pretty normal for seawater. And what happens is in December and January, it starts to go further into summer, and you get more and more algae in the water. And the algae in the water is doing more and more photosynthesis and sucking a lot of CO2 out of the water. We know CO2 makes water more acidic, so taking it out makes it more basic. And then that's a really big change from 8.0 to 8.2, and that partially happens because the seasons down there are so drastic, because you have complete darkness in the winter and 24-hour sunlight in the summer. The summer, when we say photosynthesis, it's happening day around. And then in about January and February, that algae bloom starts to die off. You start going into fall and winter, and that everything in the water is now not photosynthesizing as much, but it's respiring and breathing. And so it's now putting that CO2 back in the water. So we find that the Antarctic oceans have a seasonality to them for pH, which is pretty surprising to me, first off, because when you go there, it just always feels cold and wintry and desolate. But really, underwater, there's a whole lot of green and a whole lot of spring blooming going on that you would probably never expect if you were on the surface. So I'm going to show a little bit of diving footage, some taken by me and some taken by a friend of ours. And so the water is, again, hundreds of feet of visibility. It's some of the clearest water you'll find on Earth. We dive through one of those holes, and we have strobe lights and black and white flags on it to try to make sure you never lose that hole, because it's your only way back out. The bottom is completely covered in sea life. So this is so mind-boggling that above the surface of the ocean, it seems pretty bare, and then you duck down and the bottom is covered in, you can see some sea squirts. I think there's some sea urchins in this one. Lots of sponges and fish. So these are the fish that have evolved antifreeze in their blood, right? Because their blood is actually colder than 32 Fahrenheit. This water is about 28 degrees Fahrenheit, so about -2 centigrade. And when you're diving, you're in a dry suit, but your face is completely exposed to the cold water, so your lips go numb, which goes away after a while, but then your fingers and toes start getting a little cold after a while. So the question was, how long do we stay under? Usually the dives are about 40 minutes or so, and we do about 2 a day mostly. And it's really, you start just getting cold, and we usually make sure we come up with plenty of air left in our tank just in case. So we have a lot of safety built into the work that we do. So here's another panning shot over the bottom. You can see sea anemones, sea stars, corals, almost things that you would expect to see anywhere in the ocean. So there's just a lot of diversity covering a vast range of taxonomic groups and phylums in the water here. And yeah, it's considered, just the fact that this ocean sees no light for 3 to 4 months, like 4 to 5 months out of the year, is completely mind-boggling to me that they're living. Yeah. Yeah, the question was, what's the depth that we dive to, or the depth of our study sites? So we're limited to 130 feet, but usually our dives aren't that deep. Our typical dives are probably 80 to 100 feet. And you can see that this just goes on for as far as you can see, and there's almost clouds above you, which is the sea ice. And the bottom of it's usually a little different shapes, and it isn't as flat as the top of it. That's a ctenophore, a comb jelly, and it's probably about this big. And you can see those iridescent, it has almost an iridescence down the sides. And you'll see a lot of big open water species there that sort of come through, like big jellies and these big guys that you really don't see close to shorelines very much. And so it's a really incredible place to see huge jellies. Exactly. Yeah. Exactly. So the question was, do we avoid decompression dives? And yes, we're not allowed to go into decompression. So if our dive is at 230 feet, it's really short at that depth, definitely. And so these jellies actually have those dots on the jelly's bells are actually little critters that can only live on the tops of jellies. And they actually burrow into the bells of these jellies and make a home there and just hang out hitchhiking on them for their whole lives. You can see that the bottom of the ice there has all these different shapes in it. You'll see almost like a cave, like icicles down there. And those are called brinicles, and they form from the freezing of the seawater around it, which actually makes a super cool, super salty, sinking chunk of seawater. I said sea a lot. I think that's right. And that forms this hollow tube of ice underwater, and it just looks like that. It's just this, and everything down there can be glistening and completely beautiful. So this is one of my dives where, this is one of those seals that we saw earlier. And what they do is when we breathe, our air that we breathe out doesn't come right up to the surface. It actually gets stuck under the ice like that in these puddles of air. And these seals hang out and they actually breathe all the air that we exhaled. So that's what he's doing. He just found one of our puddles and he's going to another one, joining his buddy. And so it's just crazy to think that the air that came out of my lungs is now getting breathed by a seal in the Antarctic. So I've definitely peaked now. It's you're all applauding about that. My life is, yeah. It's okay. It's okay. And so my 3 years down there has, of course, been a completely life-changing experience for me. But it's also afforded us a really unprecedented look into the biology of these pteropods. So pteropods are worldwide, but the work on them in the Antarctic has been fairly limited. And I'm really excited to be part of a group that's been studying these pteropods and really trying to understand how this important species in this part of Antarctica might fare in the near future. And so we're discovering a lot, and I'm really excited to be able to bring this to you. And I'm now going to pass it off to Lily Simonson. So this is a photo on the right, first of all, of our field team members. So many of the people in red are in that right-hand photo. On the left is a photo of some of us with Lily Simonson. So I was really fortunate enough to meet Lily 2 years ago in the Antarctic. And as you've heard, she's been here on the Artists and Writers program. And it's been really phenomenal to talk with her and sort of, you know, when you're surrounded by scientists, to have an artist come in and ask questions that you wouldn't have imagined, to have a complete different thought process there has been really great, I think, for all of us involved. And so I'm really happy to pass this off to Lily now. Thank you.
Let's see. I think this is on, right? The Brittany Madonna mic, TED Talk mic. So yeah, I am just going to tell a little bit of my story of how I ended up as an artist diving in the Antarctic and getting adopted by Gretchen's group. So here's a slide of Umi looking at one of my recent paintings. So how this world of art and science collided for me started with the humble lobster. When I was much, much younger, first starting out as a painter, I began with a series of mammoth lobster paintings. And, you know, this is maybe 5 by 7 feet. I was really interested in sort of lobsters throughout art history, and I had my own sort of lifelong quirky fascination with them. They're so alien-like and so human. And then when I was in graduate school, I started a parallel series. And I know this sounds sort of random, but I promise you it is going somewhere. And this is this sort of a quirky story that I don't often share, but it's really how this all started, and Gretchen hadn't heard it yet, so I thought I better include it in this talk. So anyway, I was painting these moths, and they were a very different sort of series, much darker, and is there feedback? Tiny bit, okay. They centered on this childhood phobia I had of moths. And it wasn't like a cathartic thing, like a working through it thing. It was more just examining what was it about moths that were so spooky to me. And I was particularly interested in this sort of mammalian fur that covers their bodies. A lot of bigger moths are really fat and furry. And so I was really into painting the fur. And then in the middle of grad school, I was sort of looking for a new muse, looking for some more meaning in my work. And one day I opened my inbox, my email, And I had emails from like 5 different people sending me a New York Times article about the discovery of a new deep sea animal called the Yeti crab. And it was described in the article as a lobster with the same kind of fur that moths have on their bodies. And so it was this serendipitous confluence of these two, still adjusting this TED Talk mic. Okay, these two things that I thought were totally disparate were like hybridized in this one animal. Why am I, only me? I'm the only one having trouble with this.
I'm gonna move it.
Okay.
You ready? I'm like your technician.
Thank you. My muse,
You're
my
the
technician, everything.
Where's that handheld mic?
Yeah,
Maybe you like that better.
We can turn this down a little bit.
maybe that's the problem.
Yeah, we gotta turn it down. Sorry, everybody. Hang
I
on.
have my whole
Does that say on it?
roadie crew.
It's one. Roadie crew. We know she's an artist. She's
Yeah,
on tour, so-
I'm the diva
Diva
of the
diva
group.
alert. Okay, so try
Check. There we go.
Okay.
That's better.
Yeah, and if we move it down just a little bit.
Maybe I'm just more of a mouth breather than the rest of you
mouth
guys.
breathing artist. Okay, try that.
Okay.
Because the feedback
It's,
is-
you know what the problem is? I'm laughing at my own jokes too much. I'll try not to do that, but somebody has to laugh. So, anyway, so I started painting this Yeti crab. But I really like to work from observation, direct observation. That was pretty easy when I was painting lobsters and moths. But in order to observe the Yeti in the flesh, I had to reach out to the scientist that was studying it. And she was at the Natural History Museum of Paris. So I went all the way there to meet the Yeti crab, which was very exciting. And you'll see that, I'm just going to hold this. You'll see that the Yeti crab has a censored bar over its face in the upper corner. That's because the description hadn't yet been fully published, so they didn't want me to share photos of it. So I used some discretion when sharing this photo. But the lower right-hand photo is from another visit where they were a little more relaxed about me documenting the Yeti. So anyway, I got to make lots of drawings of it that I could use for my paintings, but something really cool happened. The scientist that was studying it, meeting her felt like meeting like a real kindred spirit. And I kind of realized that art and science had a lot of parallels. They're both these labors of love, where they're, you're really specialized and you're asking questions about the world and making observations and using those specific observations to answer really big questions. And I feel like that's the underlying impulse between both disciplines. And I realized that it's only in sort of contemporary society that we've diverged. Hundreds of years ago, Da Vinci is the best example, but there are lots of examples of scientists who made art, because it used to be sort of one in the same field. So I started delving into some of my heroes who were naturalist artists, and we're a little short on time, so I'll just show images of some of my influences. So I'm painting the Yeti crab, this exciting sort of mythical creature, and a couple new species get discovered. So this is a species that lives in Antarctica, actually, in the Southern Ocean, and they pile on each other around these hydrothermal vents. So I started painting that new species, visited that one in England, and then another third species was discovered that has a really crazy behavior. It looks like it's dancing. So all these Yetis were observed, 7,000 feet down, waving their pinchers over their bodies all the time. And that's sort of mysterious because it's a huge waste of energy. The theory is that they're chemosynthetic, so they get their energy from methane and sulfur that come from under the seafloor. And the idea is that perhaps this particular species of dancing Yeti is stirring up the methane and sulfur that it then harvests using bacteria that live in that exciting fur. So But it's also possible that they're just partying, right? So I tried to create like a party atmosphere for these Yeti crabs. And that's a close-up of the Yeti crab's face. This particular crab was down at Scripps, so I didn't have to travel quite as far to look at the specimen. And I started getting to know these scientists at Scripps, Andrew Thurber, who discovered it, and his mentor, Lisa Levin. And Lisa kind of reminded me that there are other organisms in the deep sea besides the Yeti crab. So I started branching out and borrowing specimens from her lab to make paintings. And I'm not going to dwell too long on these, but these are a bunch of different kinds of deep sea organisms all sort of lumped together. And then at a certain point, I had been working from preserved specimens in jars, and Lisa said, maybe you should go to sea. And I was like, yes, maybe I should. So she gave me the opportunity to join her lab on the research vessel Melville. And we were looking at organisms at about 1,000 meters down off the coast of San Diego. We had this big machine that would pull up sediment and in these cores, and then we'd sort through the sediment and pick out tiny animals. And then we were throwing the rest of the sediment over the side. And at one point, somebody on the ship said, hey, Lily, you could use that sediment as a medium for your paintings. So then I started, every day I would make a mural on the side of the ship van of something that we had pulled out of the sediment that I then got to observe under the microscope. And I'd wash it off the next day and do another painting. So I'll go through these quick. These are all little tiny critters. This is a mud owl. It's a type of polychaete worm. It's an acarax clam, a hermit crab, and a scaphopod shell. This is not so tiny. Moving into the charismatic megafauna realm with this octopus and a spider crab and a brachiopod. And then I was able to use those mud murals as sort of studies for less ephemeral paintings. So I went back to my studio and used that. It was almost like having a sketchbook, a giant sketchbook. So here's my work on display. And I began to see my role as an artist as a way to start a dialogue with a sort of different audience than these scientists I was meeting normally had access to. So obviously I can't put everything in Gretchen and Umi's papers in one painting, but I can kind of use that painting to start a conversation. So I started inviting the scientists that I was working with to come into the gallery and talk about their work. So this is a shot of one of those panel discussions in the gallery. And now I'm finally going to explain how I ended up in the Antarctic after being into the deep sea. It turned out that Andrew Thurber, who had discovered that dancing Yeti crab, he mentioned to me, Lily, I happen to have discovered this crab, but my main work is diving in Antarctica and studying these worms. And he showed me pictures of him scuba diving under the sea ice. And I'm sure others here have had this reaction after seeing Umi's images. Like, that is one of the craziest places on the planet. And for me, wanting to work from observation, I was like, I've got to see that for myself so that I can paint it. And Andrew said, well, you're in luck because the National Science Foundation actually has a program for artists. So in addition to sending all these hundreds of researchers every year to various research stations on the continent, they select one, or if there's enough money in the budget, sometimes more than one, artist or writer to embed with scientific groups and make work about what those groups are studying. So I wrote a proposal to go, and I was awarded a residency there, and I first fell in with some geologists. And we are totally running out of time, so I'm going to go through these boring terrestrial slides really quickly. But we take a helicopter to field camps. You live in a tent, And this is a buried glacier that was a feature that the geologists that I was with were really interested in. So that's me way over in the corner, plein air painting. And then, so I made a whole series based on that one particular feature. And there's me, and then I sort of fell into the allure of the glaciers themselves because they were pretty sublime. They used to think that no animals could survive on land in Antarctica, but this biologist Diana Wall discovered a few decades ago that actually, no, there are nematodes, these little worms, and tardigrades, aka water bears, that can survive in the soil. So I helped her group collect those and looked at them under the microscope and made some, there's a nematode in the foreground and some water bears back there. And then on to the diving, which was really what I was most excited about. So that's the ice edge. And I am not going to dwell on this video too long, but that's me jumping in. And I thought maybe I'd get to do 2 or 3 dives when I was there, but I ended up diving every day for a month, which was pretty exciting. So I'd go out and spend an hour diving, and as Umi mentioned, there's maybe a dozen people each season who dive there, but I thought maybe I could be at the first at something. So I had a hot dog costume. I thought, well, you know, I'm not the first person to dive in Antarctica, but I might be the first person to dive in a hot dog costume. So there I am, just a little performance art side project. But I was really there to paint. So I had a little studio on station. So there's me in my studio. And I got really, again, sort of seduced by the underwater landscape. So that's a painting I made of a seal diving under that beautiful stained glass is a great description of what the sea ice looks like, that glowing, textured sea ice. This is a submerged glacier. So that opaque white ice is the glacier, and then that yellowy stuff at the top is that 6-foot thick ceiling. And then these are pressure ridges, an area where the sea ice gets a bit thicker and you get these cave-like formations. Here's a little brine channel. And then on all my dives, I would collect things that I wanted to paint and bring them back to the research station and keep them alive in the touch tank and then use them as models. So it was a huge step forward from working from preserved specimens to then collecting my own specimens and keeping them alive. And these are nudibranchs, so I collected those. And here's a nudibranch painting in progress back on station. And then this is the finished painting. They're like a type of sea slug. And then this is the cousin of the pteropod that Gretchen's group studies. This is Cleone. It's another species of pteropod, and they're called sea angels. They don't have that shell, but they have that wing-like appendage. And then I met Gretchen and fell in with her group and got to help them collect pteropods, and just totally became obsessed with the beauty of those pteropods. So this is a portrait of a pteropod, and then you can see the real deal painting over there. You don't need to see this slide for too long. And that's sort of a baby-sized painting compared to what I usually do. So this is me working back in my studio in Oakland. So I started these paintings in Antarctica, then I shipped them back to California and finished them there. And then this is my most recent exhibition. And then I did my usual thing of hosting a panel discussion with Joe Levy, the geologist I worked with, Rob Robbins, who's a diver who runs the diving program and has done 2,000 dives, more than 2,000, under the ice in Antarctica. And then Gretchen, of course, was there. So they all talked about their work. And it was great. And I'll wrap this up by skipping ahead. I get to go sort of full circle. I'm getting ready to go back to sea again on the Atlantis, and we will be visiting Gettysburg off the coast of Costa Rica. So that's where the dancing Yeti lives. So I feel like I've peaked diving in Antarctica, but I'm pretty excited about possibly diving in Alvin to like 7,000 feet down. So wish me luck. That's the next adventure. So anyway, thank you all so much for coming. Oh, there.
That's okay. Here, let's have this. It's this one. Yeah. Are you happy with that? Sure.
Yeah.
Yep.
You can turn this off.
Okay, everyone. We're going to do questions. So here we go. Ready? Logan's going to run the mic. Oh, I just booted that. There we go.
When you talked about the seals collecting the air from the exhaust from your regulator, was it an air pocket that they were able to grab the oxygen from, or was they actually able to synthesize the air out of the water?
No, I think they were just going for air pockets underneath the ice. And I think they know to do that naturally. I think you can get naturally forming air pockets under the ice. Sorry about that. The question was, do the seals actually just sip on air pockets under the ice in that video? And yes, that's probably a learned behavior from maybe some natural air pockets that probably form.
Great presentation. Thanks, you guys. Yeah, I'm interested in those experiments where you simulated different CO2 levels in the seawater. And I'm wondering, does that correlate to the CO2 in the atmosphere at all? And did you see a red flag happening with that creature and maybe the ocean in general at any particular CO2 level?
Sure. So yes, the CO2 in the water has a lot to do with the CO2 in the air. So of course, the first thing that happens is CO2 in the air sort of mixes and diffuses into the water and sort of forms that equilibrium. And then certain processes in the water can sort of take over from there and potentially change the CO2 in the water. So the pattern that we saw over the seasons was probably caused by some interplay between the biology, changing the CO2, so the photosynthesis and respiration I mentioned, and also the fact that for part of the year, there actually is there's a barrier, right? So it's frozen over, so the CO2 in the water can't actually equalize with the CO2 in the air when there's a cap over it with ice. And so it's probably some sort of interaction between the physical cap over the ocean and the sort of biological processes happening. And we didn't, I think, necessarily see a tipping point per se, but we're sort of still investigating some of the data that we've collected and looking into some other aspects of pteropods that we didn't discuss today. So I'm hoping to have those questions answered soon.
What is the highest CO2 level you simulated in the lab?
Sure. I'll take you off the hook. I'll take Umi off the hook for this one. So right now the atmosphere in the northern hemisphere on average is about 400 plus microatmospheres CO2. That's kind of going up a tick or two every year because of our emissions. In the Southern Ocean, there's almost a perfect equilibrium between the atmosphere and the ocean, so it's usually somewhere around 400, but the cap of ice, I think we actually sequester CO2, so a lot of times we find it at 450 or 500. And when the ice comes off, there's some loss of the CO2. The phytoplankton pulls it down, takes it out of the water, alkalinizes the water. But what we see when we do our lab experiments is that the level of CO2 that we used was about 900 to 1,000 microatmospheres, so doubling what we see right now, a little bit more. But that's what we call worst case scenario. We change nothing about the way we emit carbon into the atmosphere. Multiple coal plants go back online or come back online, and we continue on a trajectory such that by 2050, we have incredibly high levels of atmospheric CO2. And as I said before, I don't think it's going to happen, but although we don't see a place where pteropods just completely tip and die, we do see that if we get to that 900 PCO2 level, their shells start to dissolve. And it's pretty clear that present-day genotypes are really stressed by that. We look at their gene expression and they are not happy. And unless they're able to adapt rapidly to future ocean change, we stand to lose a part of the food chain that's kind of a drag, which is why I really advocate for, learning where we see vulnerabilities in nature. We don't have infinite time or infinite money, and so we want to look and see where we can expect trouble for humans. So, any other?
Thank you guys for coming.